So it's a great pleasure for me to be here. I'm a representative of the group which is here, a group originally started by Dimitar Sasolov, who's an astronomer, and Jack Shostak, who is an RNA guy, and then others have grown in. And what I would say about us is that each one of us has strong opinions which happen to have almost no overlap. And so I fight endlessly with these guys. We get along very well, but we don't actually know the answer. And what is, to me, so exciting about this area is that much of science proceeds by discussions of development of a topic which is already, in a sense, understood. And as Eric said, it would be nice to put this on a footing where one can develop well-defined hypotheses and go forward. We're not there yet. So what I'm going to tell you is less about what I don't know and think needs to be known than about what I do know, although I'll tell you some things that might be a start to what we do know. So let me start with some broad questions, since as the first speaker, one gets to talk about things which have almost no substance. Here's one. Where did we start? Where did life come from? I am a bottom-up person. I'm interested in how you go from whatever it is that fell in from space and was produced by geochemistry to the first primitive things that might have become cells. What comes later is very interesting as well, but I'm specifically interested in this question. A second question, and I'm not sure whether this is on. Can you hear from the back? Okay. At the beginning, we know that the planet was a real mess. It was the end of the period of heavy bombardment. There was all kinds of stuff going on. You would think it was an ideally bad environment to create something that was delicate and organic and just hanging on to being organized. How did that happen? And I don't think that this process of going from a chaotic environment into an ordered cell is at all understood. A third question, which is really interesting from the point of view of trying to decide what we're doing, is how do we think about life? And the question about life, in a sense, is how do we know when we've succeeded? If we study this area, we're interested in the origin of life. How do we know that we're there? How do we know that we've got life? What is the first thing that's alive? Not an easy question to answer, I think. And so that's an interesting one to consider. And I have to say, just to give you a sense, that I think that it's a perfectly legitimate exercise to think about life as being a form of flame in which, instead of generating heat from the combustion of methane and oxygen, you generate from the combustion of oxygen and glucose. It's just a set of interesting chemical reactions. Are alive and not alive different? That's an interesting question, and one which is important if you're interested in the origins, because life and death are the opposite sides of the same sandwich in a sense, and you should be able to understand the two. And then finally, a question we will talk about and others will talk about, which is the explosion of fascinating stuff that's going on in planetary astronomy. If life is inevitable on any planet with water, then there must be life everywhere, at least sparkles of it, and that changes our view of our place in the universe. Now, let me give you a brief history. This is going to, if some of you are professional organic chemists, you should find this infuriating. I say organic chemistry primarily because life is largely, at least in terms of mass, an organic phenomenon. Inorganic chemistry is important, and physical chemistry is important in thinking about it, and physics is important overwhelmingly in the overall arching thing. But let me give you a history of organic chemistry. It started by making complicated molecules, first simple ones and complex ones. And these molecules are trapped metastable states, by which I mean that all of us at thermodynamic equilibrium would be CO2 and water and nitrogen N2. But we don't burn because we're trapped. Okay, got it. Recently, the last 20 years, there's been a shift toward non-covalent chemistry and molecular self-assembly, These are equilibrium structures. The bonds that hold things together are weaker, and they're interesting too. You have a pretty good grip on these two, and neither one of them is sufficient to explain the origin of life. So what one has to do is to go to the next step, which is dynamic systems. And chemistry, when you're out of equilibrium, when the phenomena that you're looking at require the flow of energy and the dissipation of energy through the system that you're looking at, and what's interesting is the result of dissipation of energy is something we understand very, very poorly. So it's a wonderful area from that point of view. And why is it that chemistry hasn't gone this route? And I'll give you a sense. Here's the result of complex chemistry. Here's self-assembly. And my mother, who was a wise and difficult woman, used to say that, why haven't people gone on to the other thing? And her view was you can never change anyone's mind. You just wait for them to die. This was said in a somewhat more fashionable way by Mr. Pauli, who said that science progresses one funeral at a time. So we're waiting right now for the people who do complex organic synthesis and molecular self-assembly to reach my age and die. Themes. What came before? And here we begin to get into some of the interesting subjects. One of them is, as I'll come back and discuss in a bit, there are a number of different ideologies in this area, not overwhelmingly connected. One of them that's been very, very successful has been work that started really with, I guess, Orgel, but was Orgel and people of that sort, Uri Miller, but reached a very high point with Albert Eschenmoser and then with Jack Szostak continuing, which is to start with simple molecules, HCN, methane, ammonia, carbon monoxide, carbon dioxide, so on, and do prebiotic chemistry. That is to show that plausible reactions starting with simple materials can make the kinds of molecules that you find in life. It's worked very well. The question of how relevant it is a question which you can discuss, because much of this chemistry, all of this chemistry goes on in organic chemistry conditions, in flasks with good Swiss postdocs running the reaction, and that doesn't detract from the chemistry, it just raises questions about plausibility. What had to happen at that point is that chemistry somehow organized itself into networks. That is the beginnings of metabolism. We don't know how that happened. We do not know how that happened. And that's a key point. So there are ideologies. And one ideology is the ideology of organic chemists who make complex molecules. And John Sutherland is an expert at this, and he does it fantastically well. And then you assume that the rest takes care of itself. A second ideology is the one that starts from the biology and works backwards. And my friend Jack Szostak is big on this. He says, assume that with enough oceans and enough billions of years, RNA spontaneously emerged. Well, that makes it easier. And the question of how you connect these two, the molecules and the precursors, comes to this issue of the networks. And so we have networks, we have molecules, we have precursors. But there are some parts that are actually missing. And to give you a prejudice, I, by professional background, am really someone who goes through the steps of quantum mechanics to statistical mechanics to thermodynamics. I think this is one of the great triads in natural science. And one of the things that we're missing here is understanding not what could possibly have happened, but what could statistically have happened. What is actually plausible statistically? If you have molecules and you put them together, will they ever spontaneously form networks? And I'll come back to that in a bit. And then we go on with other things, but eventually one has to get to this big trick of evolution, which is molecular memory and expression, and the two pieces that go along with it, and a key element which is not well understood at all, which is the emergence of enzymatic catalysis. Because networks in biology are really networks of catalysts, not networks of molecules. So then, let me summarize this part of things this way. There's the chemistry forward part. This has gone extraordinarily well. This has gone well in academic laboratories. There's the backward way, which starts with DNA, RNA, we all agree is an important component and comes from somewhere. And what's missing is this part in between, which is where did metabolism, networks, catalysis, where did that come from, and how can it possibly have happened? And to be perfectly frank, I'm a professor. And professors have the skill of being able to take any subject and talk about it knowledgeably. I have no clue. I do not understand how this happened. So it makes it a great problem. Now I want to give you two pictures to think about. These are very simple, but they're actually pretty profound. You all have seen this. You take coffee, you pour something into it, it spontaneously mixes. Nothing to think about there, or is there? If you look at it the other way around and assume you go from left to right, and the answer here is, well, you're never going to have a cup of coffee with mixed coffee and milk, spontaneously spit out milk and form that. We know that. Why is it? And whenever you ask the question why, you run into problems in science. And you say first, well, that's not fair, because what you're doing here is something in which, in this previous picture, what happened was the coffee fell in. As the coffee fell in, it mixed. As it mixed, something happened entropically, and there was heat generated from gravity and potential energy, and you can't get those two back. But, you know, my view would be, okay, that's fine. This still isn't going to happen if I put it in boiling water. So I've got lots of energy, or X-irradiated or whatever. I've got lots of energy. So it isn't the energy issue that's the important issue. It's the entropy issue. Now, there's always this question that is raised in discussions of quantum mechanics, where if you talk to people about quantum mechanics and you say, do you understand quantum mechanics? And they say, yes, we understand quantum mechanics. You know they don't understand quantum mechanics, because quantum mechanics is not really understandable. And the trouble with statistical mechanics is that although it's nothing more than throwing dice, it's really hard to understand when you get to big numbers. And so the question of why this doesn't happen is not the trivial thing that you might think it would be. There's lots of energy around in principle, but the mixing, the question of what you do when you take 10 to the 23rd of something and mix it with 10 to the 22nd of something else, is quite profound to think about in the context we're worrying about. Because you can make the molecules, but they're in an ocean. And so how do you put them together in such a fashion that they do something? And this is the more important problem. We know how to convert this into this. And the question is, do you know how to convert that into that? Because everything that's here is basically here. And it's just in a different form. It's a trivial question, but it's not at all trivial, and it's not just a question of chemical reactions. It's not a question of RNA. It's something else. And the something else to me is what we don't understand, even conceptually. Now I want to ask you another question. And this has to do with the subject that everyone here understands. Maybe. I don't, but everyone else probably does. And that's self-assembly. And with molecules, we understand self-assembly pretty well. What one has is two pieces of this sort. They're going to self-assemble. When do they self-assemble? You need to have two and only two things, really. You need to have an attractive force which brings them together. This is distance, this is energy. They come closer together, the energy goes down. But then you also have to have a repulsive force or energy, which when they get close enough, pushes them apart. And if you have those two, you add a repulsion and an attraction, you get to something that looks like this. And what that says is, so long as there's thermal agitation, the system will come together until it reaches this bottom here. The agitation has to be sufficiently small that it doesn't get kicked out, but so long as the agitation is small enough, it'll come to rest down here, and that's self-assembly. Got it. Happens all over. But how do reactions self-assemble? Because that's what we have to talk about if we're going to talk about networks. We know that there are reactions that occur, and somehow those reactions have to come together to give a network in which the reactions are talking to one another, not the molecules talking to one another. You're talking about the self-assembly of a non-material thing. What does that mean? I want to give you just a couple of examples of background in this to give you what at least bothers us or bothers me about the difficulty of that problem. This is all familiar. This is work from the 80s that we did, and other people have worked on the same kind of thing, that show that you can get all kinds of interesting molecular self-assembly by using hydrogen bonds of the sort that make DNA and RNA and other things, proteins. In some ways, not a problem, although I should point out it is a problem in other senses. But I want to show you an example of dynamic self-assembly. This is the simplest system that we can think of. What one has here is simply droplets of air, bubbles of air, coming through a T-junction, forming bubbles here, and then the bubbles expand into a channel that looks like this. And if you look at this, these things come through, and look how they self-assemble. Stunning pattern. The physical chemistry of this, the physics that underlies it, is extremely straightforward in principle. In principle, it's the Navier-Stokes equation. But having said that, would we have predicted that it does this? Can we explain it? Yeah, sort of, you can explain it. But it's still a surprise, and this is so much simpler than what one is thinking about when one thinks about a dissipative system in life, that it's almost not the same ballpark. And let me show you another example. And I think I have to run this mechanically. This system is, the blue beads are nothing but little nylon spheres, and the white beads are nothing but little Teflon spheres, and they're in a dish. And we shake the dish, and the fact that shaking is erratic is just non-sync between the camera and the dish. And I want you to watch what happens. And this is a true dissipative system. You're putting in energy. And you notice they sort of rattle around, but you see sort of little parts of things beginning to organize. And then pretty soon you find larger pieces of things beginning to organize better. And then what you find is, after a little while, the whole thing crystallizes. Now, what's happening there? And after a while, I won't go through it all, but what happens after all this becomes basically a perfect crystal. And what's happening is a dissipative process in which, as these beads shake, the blue beads spontaneously trade ions with the surface in which they're resting, tribocharging to become negatively charged. The white beads become positively charged by chemistry that's not understood at all, really, and then you get a two-dimensional Coulombic crystal. Again, I put these up not because they're relevant to life, but because we can barely understand this level of complexity. All right, so replace my little dish with the shaking beads with this, and it gets to be a harder problem. And so there's lots to be studied here. And a final puzzle in this introductory piece, and this to me I think is one of the more interesting problems in all of science right now. We know that a cell, whatever it is a collection of molecules, and we know that a molecule is not alive. Everybody would agree with that. We know that reactions between molecules are not alive. I don't know how to talk about a reaction as being alive. We know that cells are nothing more than molecules which are not alive, reacting, which is not alive, and yet the collection is alive. How did that happen? What happened there? And we don't know. And by the way, if you like extrapolating, I would make the argument that thinking is nothing more than reactions of molecules which are not thinking in arrangements which are not thinking. And so thought is the same thing. And this is my sort of conceptual argument that says that a flame is, after all, nothing more than an organized structure which is doing something, getting off heat and light based on combustion. If you reorganize a flame, it becomes you. And if you organize it a slightly more complicated way, it becomes a flame that thinks, which is you, certainly not me. I barely think. So these questions are very, very interesting in the sense we truly, or at least I truly, have no idea how to put this together. This is not trivial science, this is good science. So let me go back to my questions. Where did life start? And are there other kinds? And I haven't said anything so far that says you couldn't have life based on a lot of other different kinds of chemistry. I have said so far that it's chemistry. Was the start deterministic, or did it just happen by accident? A very important question, because we don't have any way right now of saying that maybe we are the one place in the universe in which it just happened that enough oceans with enough time produced a protocell. But we don't know. And this comes into statistics and probability, which is an area that this field has resolutely ignored so far, because it's an uncomfortable area. How does dissipation allow chaos to come in? And one of the interesting questions here was it chaos? Or in dissipative systems, is there a physics that we haven't really thought about? Is there something new, a new principle here? And I don't know what it is, but that's what makes it interesting. People didn't know about quantum mechanics in 1900. There was just one or two little things that didn't fit that forced the invention of quantum mechanics. Are they separate, alive and not alive? Is a flame alive? Is a city alive? I happen to think that cities are alive, but that's a different issue. How similar was life then and now? Obvious question to thinking about. And the interesting issue here is that we look, I think all of us look at what we see in ourselves and say that that reflects in some way some trace of what was there at the beginning. But maybe then was different in some way. You say you're just waving your hands, and that's absolutely true. I'm waving my hands, but you don't want to miss things, because quantum mechanics, to take an example, did not come as an extension of Newtonian mechanics. It was a fundamentally fresh start. And are we missing the fresh start here? Is there more life than just the set of molecular precursors? For example, the flame. And then I'm going to come back to this. How do we know if we've been successful? So, we know that the business of life is to make more life. That seems to be what goes on. But it's easy to mistake one cell becoming two and two becoming four for this. And what goes on, the machinery inside, we sort of understand this. What we do not understand at all, we don't even understand how this operates. Homeostasis in the cell is still pretty much a mystery in large part. But if you ask where it came from, we don't know. So to me, and this is my prejudice, this is where the core of this problem is. Where did that come from? How could it possibly have happened in the chaotic environment of the early Earth? And I would put just one more final detailed point on this, and Jack Szostak and I argue about this endlessly. The wonderful thing about the DNA, RNA kind of story, and Jack makes a big point of this. He says, let's assume RNA. Okay, we assume RNA. And then RNA has the virtue that it is both a catalyst and something that looks like a memory molecule. So RNA can do it all. That's the basis of the RNA world story. The difficulty with that is that RNA is actually not a very good catalyst. It works for nucleotides and things like that. And it's actually not a very good memory molecule. So the fact that it resembles DNA, and it certainly is a piece of the ribosome, is all fine. But then how do you get to something that looks like a primitive ribosome? If you had no machinery for making proteins and no knowledge of where RNA came from, then how do you get to a starting ribosome? And it's these pieces that are missing. So one of the things that we've been talking about in the group, and it's a key part of what I'm going to say, is that there may be ways of doing this chemistry of making proteins and making other things that are not based on the system that we have right now. And there is one, I think, interesting kind of example, which is non-ribosomal peptide synthesis. This is to do with the so-called thioester world, in which instead of making fairly complex oligopeptides, one by a ribosome, what one does is to make them by another piece of machinery in which a series of thioesters react with one another. And although in bacteria, this machinery is pretty well organized in proteins, in principle, you could at least imagine that it might have been organized on other kinds of matrices. And this might be a sort of step. And although I'm not going to talk about this, I am going to use thioesters. And why? And this is one of the things I think we chemists want to look at. Thioesters are actually pretty remarkable. This is a thioester. And it has a very important characteristic, which is that it will react with a thiol, that is RSH, to do an interchange of this for this, and hence of the products that come from this. It will do that in quantitative yield. Why is that important? And I think there are many arguments that say the reversibility of these reactions is important in generating the arrays which allow selection, which allow optimization. I mean, this may not be true, but I think that a key component to prebiotic evolution has to be reversibility. And this is a remarkable reaction in that this functional group is beautifully reactive, but unlike other activated esters, it does not hydrolyze. So, it does, but the rate constant for amide formation relative to hydrolysis is in the order of 100,000 at an accessible pH, which is about 8. I don't know another reaction like it. And that's one of the arguments for the de sort of approach to things. So what are we doing? Our interest is in trying to design networks that enable us to learn something about complexity on a scale that is similar to that of metabolism. Similar meaning we like something that looks the same order of magnitude complexity as the Krebs cycle. What we're working on is simpler than the Krebs cycle, and I'll show you in a minute, but we like something that's roughly that order of magnitude. So our choices. We're going to make amides because they're important for proteins and peptides via thioesters. What we're missing, and I know this to be true, is we're missing catalysts that provide control. And that is the key to metabolism. It's not the reactions per se, it's the catalysts which turn one another on and off. We need simple organic chemistry, so nothing complicated, no Swiss postdocs. We are going to be using as our product not the chemical reaction, but rather oscillations, because that's a complex behavior and it's pretty easy to follow. So you need, as we were discussing earlier, we need an observable. We're going to include a mass transport step, and I can discuss this with you as a while, but our major shortfall is that we don't have good approaches to catalysis, and catalysis, in my view, is one of the key things that we're missing in this whole area. So Krebs cycle, the only point to make here is that it does the same kind of thing that we're interested in. It's a simple input, which is a thioester. And what we don't have is that every one of these steps has a catalyst. Some of the catalysts look very much like things that could come out of prebiotic chemistry. All of these things look that way. But we don't actually have ways of connecting the prebiotic chemistry to these sorts of reactions. So in principle, it looks good. In practice, the activities of the little iron sulfur clusters and iron manganese sulfur clusters have to be modulated in ways that we don't understand how to do right now. And things like an imidazole phosphate, simple piece, but we actually don't know how to make imidazole phosphates. So let me tell you about the experiment, and I want you to think of this in terms of an analogy. And the one that I keep coming back to is life a flame? Well, what's a flame? You put in an reductant, you mix it with an oxidant, so methane and oxygen. There's an organized structure. The organized structure is probably with methane, oxygen, it's in the order of 10,000 reactions, something like that. We don't really understand it. And anything more elaborate than methane, we don't understand at all. There are no models for those systems. But these two, oxygen and methane, produce an organized structure which does something. It gives off heat and light and converts methane into carbon dioxide and water. And if you want to, you can put in various sorts of machinery here to make it to do pretty interesting things. I won't go through this here. We say a cell is a structure, looks like this, into which goes nutrients, glucose, oxygen, some other stuff. And what comes out of it is waste, and what it does instead of generating heat and light is to make another cell. You say, quite correctly, that is an analogy so loose and so sloppy that only a decrepit geriatric old man would come up with that kind of thing. Got it. Geriatric decrepit old man. I qualify there at least. We're going to add one component to it, which is what's called by chemical engineers a continuous stirred tank reactor. And this is just basically something in which is a reactor, a little container. Reactants can go in, products can go out, and there's a stirrer. You say, what does that have to do with anything? And that is, it's exactly like this. Reactants go in. Here, there's no mechanical stirring. Well, there actually is some, but that's not the key issue. This is smaller, so diffusion allows everything to mix. Here, because it's a little bit bigger, it's more convenient to have a stirrer, but it's not absolutely necessary. So I'm saying a cell is like a continuous stirred reactor and like a flame. And here's the actual system, not important, the details. Here you have a mixer. We feed reactants into it. We have it thermostated, not important. The products come out of it. We analyze them, and that's all that happens. Now, what's the chemistry? I put this up because it's like taking a typical organic chemistry class and putting up differential equations. Everyone looks at it. For about 7 microseconds, their mind goes blank and they start thinking about something else, dinner. And so I'm not going to spend time on it. The only issue is that what happens here is that you take a thioester, this one, you convert it into a product, which is an amide. Let me find it here, an amide. And every time you do that, you make two molecules of the reactants that are involved. So it is a designed auto-amplifying reaction. And the details, I'm happy to go through it with anybody who's interested, but there's a point which is actually a quite interesting point here. You can describe all of this with a set of about, I think it's 6 or 7 coupled differential equations. I should say as an organic chemist that organic chemists do not do coupled differential equations. We just don't do that. And the trouble is that even if you do it, you can look at such a set of reactions and work them through, but it doesn't actually tell you very much. So we spent some time worrying about what language we use to describe that. And what we've settled on is this, and I recommend it to you to think about when you're thinking about the cell. We use what's very common in electronics and also in chemical engineering, which is to use something called control theory. And in control theory, you ultimately get around to differential equations, but you think about things rather in terms of functional blocks. So all that's involved in what I told you is that we have reactants, and they're flowing at some rate into the reactor. This is the continuous stirred tank reactor. Some of these reactants cause a trigger. Well, actually, let me do this. The auto-amplification reaction is the key step. In an auto-amplifying reaction, you trigger it, and the rate goes up exponentially. That's the characteristic of auto-amplification. So what we have to have is a trigger for this, and we can design the trigger. The trigger sets off the auto-amplification. The auto-amplification eats all of the reactants, and then we wash it out and reload it by this continuous stream. So that what you have to do, all you have to do in a sense, is organize the rate at which reactants go in, the concentration of the reactants, and you will see various behaviors, and the one that we see is an oscillation that looks like that. And the reactants are all reactants that are plausibly present in prebiotic chemistry. This, arguably not, but the rest of those could certainly have been there. So limiting cases, one is that if we flow stuff too fast or too slow, you just have steady state. But if you do it the right way, you have oscillations. And so we observe the oscillations. This is in the order of the degree of complexity of the Krebs cycle. And it shows some really interesting behavior. This is all now occurring spontaneously. One of them is that if you go forward, that is, you increase the flow rate in one way, you see multiple steady states in the product that we're looking at. Here's a steady state, there's another, there's another, there's another. But if you go backwards, this is what you see here, if you go backwards, you see totally different behavior. That is, you increase the rate constant for flowing the liquid into the reactor. You see stationary states, but if you decrease the rate going backwards, you see no such stationary states. You see something that's totally different. This kind of complexity is the kind of thing that one can imagine using for making something more complicated. Here is oscillations, and the one problem with this system, I should say, experimentally, is that the oscillations are slow. It's one every hour or so. So we have to make them faster. But if you look at the space velocity, which is the number of times per second that the contents of the reactor are exchanged, and these are 10 to the minus 4 or so. When you get down here, you have a kind of stationary state in which you turn the system on and it will do that. That is, the oscillations will go away. In here, you have nice oscillations, and you go over here, and the oscillations go away. And it's not over a very wide range of space velocities. This is a pretty delicate system. And I'll show you why that's important in a minute. Now, you say, let me go back to this. I say that this kind of complex behavior only occurs in a narrow region. That doesn't sound like the kind of thing that one wants to have when one is thinking about a kind of chemistry that might model something that was involved in developing life somehow, a section of metabolism. So it looks too fragile. And anyway, the environment then did not have clean reactions. It had variable temperature, it had variable flow rates. Maybe all of this is just nonsense. And so we've set out to look at this subject of robustness. And so this fits in with something which is, I think, a very interesting question, which is Darwinian evolution. Now, an argument with Darwin and evolution, which we'll come to in a minute, is finches. And finches which adapt themselves through evolution to be specialized for picking pine cones out of bark and things of that kind. So specialization, which led to the fastest cheetah or the fastest deer or the best beak, optimized performance. But it's not absolutely obvious that that's the right vector at the beginning, because you don't have to worry about competition with species if there were no species to compete. It's just not an issue. So maybe it's a different story, and what you want is something that's the most durable and robust system, not the most specialized system. And we wanted to see if we could find any evidence for that one way or the other. So what we're going to do is to do the following thing. We're going to take the system that I just showed you, and we're going to vary things. So here are the finches that I just talked about. And what we've done is simply to take, instead of one set of reactions, I'm going to take two set of reactions. I'm going to add two thioesters and see what happens. So I've got a mixture, a minimal mixture. And in doing this, we've seen something which is quite interesting, quite intriguing. And let's not worry about the details here other than to say that we have different structures than amino acids. And what these truth tables here show is that if we take a system that contains alanine thioester, glycine thioester, with glycine and glycine, imagine we're doing this with two systems, and we just mix glycine and glycine, so it's just glycine. It doesn't oscillate. Alanine with alanine always oscillates, but what's important is that if you mix alanine with glycine, it still oscillates. So what we see is that if you have a system that's oscillating, it can entrain a non-oscillating system. So it's actually more robust as a mixture than otherwise. And even more remarkably, if we take a system of glycine, which doesn't oscillate, glycine, and a system of this amino acid, which doesn't oscillate, neither one of them oscillate, and we put them together and they do oscillate. So it's a very primitive result, but what it says is that in this kind of evolutionary issue, it actually might be the case that mixtures are preferred. And in fact, you can make an argument in a number of circumstances that Darwin may have had the right answer, but not quite the right question. And that is, when you have species that are optimized, maybe you want specialization. But for robustness, what you might want is a mixture of species, some of which do really well in some circumstances, and some of which do really well in other circumstances, and which cover the range, but don't individually, are not individually the best for some particular circumstance. And I think that's important because it gets around, in principle, some of these problems of how do you deal with mixtures. So, what I just said, but now I want to raise a question which I can't answer, which is the one that's here. This is all very interesting chemistry, and it does tell us something about dissipative systems, but is it relevant to this? And my good friend Jack Szostak would say, this is total nonsense. It has nothing to do with anything. And I would say to my good friend Jack Szostak, RNA is a wonderful molecule, but you're never going to get it. So we tend to look at one another across the table and go have lunch. All right, so should I quit at this point?
We have about 17 or 18.
All right, well, let me quickly go through this because there's an important point that I want to raise. And that is that, how do we know when we've succeeded? How do we know what's the first sign of life? And I don't have any idea. But there's an interesting question that I can ask here. What is life? When I'm a chemist, and chemist to chemist, the world starts with electrons and nuclei, and you build everything that way. When I go to my friends who are physicists and I say, humble chemist, I'm a humble chemist, what's an electron? And I get the same answer every time, and that is they look at me with a certain condescension and say, you can't ask that question. You cannot ask, it is not a legitimate question to ask what an electron is. You can only ask what an electron does. Different question. And so we may not be able to ask the question of what life is, but we can ask what life does. And it's a different way of thinking about the problem. So these are all things that we know. Bounded and compartmentalized, we can do that. Cyclic, yes. Adaptive, yes. Memory, we don't quite know how that's fitted in the beginning. In water, let's assume it. And then the three things that I think are most interesting are in red. It's out of equilibrium and dissipative. And what does that have to do with what goes on here? It's alive, yes. And is it emergent? And what I mean by that, without getting into the semantic swamps of what emergence means, is there some principle in statistics, in probability, in thinking about robustness and all the rest of these things that we don't get? Are we assuming that we know completely everything that needs to be known to answer this problem? And we know what we know, so the answer is always, to this question, always going to be yes, unless you're cautious, in which point you say, quantum mechanics in 1900 didn't exist, and that's important. So let's not spend time on that other than one question. What are the limits to reductionism? Good question. But think about this just a bit. Are alive, not alive, and dead, partially dead, other than not alive, how do we think about those words? It's very interesting. We don't actually have a word for a stone. We have a word for alive, and then we define everything by being not what you are. But there might be lots of states that are not what you are, but we don't have any words for any of them. And what you don't name, what you can't name, you can't think about. So let me just ask, which of these are alive? Is a frozen embryo alive? There are no reactions. Megacity? Megacity, to me, has all the characteristics of a cell, but the organism doesn't have molecules, it has people. The World Wide Web without people, World Wide Web with people, and one of these should be with and without. The Internet of Things in 2080, I chose that 50 years from now. So is that going to be alive? We don't know the answer there. The Internet of Things, a virus inside a cell, all the rest of those things. Could there be life that's completely different from ours? And I think we should think about that in terms of something that looks like this. And there's just one or two more diagrams I want to show to finish off. Suppose we think about some kind of metric here, complexity or complicatedness or something else, proximity to life, and then aliveness. Is there a continuum there? Do you go smoothly from this to fully alive? Or is it something that looks like this? And if you look at any variant in complexity, what you find is that there are all kinds of responses between an input function and output function in I have no idea what dimensionality of space. Binary, it's just not alive, and then it's alive. Here you might say, well, it's not alive, and it gets more alive rapidly as it goes on. But it's really these kinds of functions that I think are most interesting, these bifurcations in multiple dimensions. And in this kind of thing, you have something become more complicated, and at some point, something happens. And the something happens involves a transition. But then you change it and you move it down, and something else happens. Those of you who know about complexity know about this kind of thing. But I note that as I have gone through a process of being a fertilized ovum. I got more complicated. I became a little piece of meat, which is called a baby, and then more complicated, more complicated. At some point, I'm going to die. And when I die, when I'm dead by everybody's definition, my toenails and my hair will continue to grow for a long while. Now, what part of me, at what point do we define me as not alive? And what happens is the brain goes off, the heart goes off, you stop delivering tissue, metabolism starts. What did I just say? Metabolism stops. And that, I think, is probably a greater criterion for alive or not alive, but I'm not sure exactly how to think about it. So, just two final slides. What are we missing? This community, in my view, doesn't know very much about thermodynamics, statistics, probability, or information. And I think it's important. Catalysis, I would love to make progress in catalysis, but I haven't had any good ideas yet. Dissipative systems, the Leventhal paradox. How many of you know what the Leventhal paradox is? It's a good illustrative example. Maybe we'll talk about it at the end if somebody asks a relevant question. So somebody raise their hand and talk about the Leventhal equation. Combinatorial chemistry has been a complete failure because we don't know how many things we need to have. All the rest of these kinds of things. What I'm saying is we have organic chemists doing good organic chemistry. We have biologists doing good biological chemistry. But we've completely left out so far the physical chemistry and physics to provide concepts along the way. And then here's stuff that you can just read as you think about it. Not important enough to go through here. And that's really what I have to say. But what I would conclude with is just a short statement that if you look at what we know so far, we have wonderful pieces, progress in a number of areas, but we don't actually know whether the areas where we have progress or progress in a direction that's leading us to something that we can put together into the schemes that make the first proto-living thing. And we don't even know what the first proto-living thing would look like. So we don't quite know how do we know when we're there? We need to think about how we know when we've reached there. It's a wonderful area. It's the most interesting area of science to me right now in molecular terms. And I'm glad we're all working on it. Thank you for the invitation to be here.
So, wonderful talk. I think that polymers that avoid the Leventhal paradox are probably the most interesting ones to go after. So could you talk about the Leventhal paradox?
Let me tell you, thank you so much. I mean, I owe you a drink, a small drink. The Leventhal paradox is something which addresses the following question. It's very instructive to me in what it teaches. You look at a protein, and here's the sort of argument, and you say, well, maybe proteins were evolved by randomly putting together amino acids into pieces that became big enough to be catalytically active. You just put them together. And let's take a protein with 300 amino acids and do the statistics. And you take, in the first position, you can put 20 amino acids. I mean, I'm oversimplifying, but 20 in the second 20 in the third 20. So there might be 20 to 300th combinations. And then you do the experiment of making one molecule of each of those 10, 20 to 300th proteins. You pack them at a density of a crystal, and you ask how big is that piece, and that piece is bigger than the known universe. So whatever happened, it wasn't that. And why that's interesting is it says there's another pathway through this complicated phase space. Probably you made little pieces, maybe that formed helices or formed beta-pleated sheet-like things, and the pieces began to aggregate some way, and there was some pathway by which evolution selected along the way. And I think looking for those kinds of principles, simply rather than saying, look, enough oceans and enough billions of years, and we'll get there, I think that won't work, and we don't know what the guiding principles are. Is that a satisfactory answer?
What life does is to hydrogenate carbon dioxide. So that's its job. And if you take that diagram of yours with the Roche diagram, you find right in the middle not only the Krebs cycle, or the reverse Krebs cycle perhaps, but also the acetyl-coenzyme A pathway. That seems to be the initial pathway. And George Fuchs pointed out a long time ago that gluconeogenesis predated glycolysis. So if you build everything up from CO2 and hydrogen and methane, shall we say, and nitric oxide, then you can have a third spot at your table for lunch.
I think that's probably true, and I'm willing to take anything that I can convince myself could happen in primitive conditions. Now, one of the problems with hydrogen is there certainly was hydrogen in certain regions, but not a lot of it because it tended to boil out. And the reduction of CO2, what?
Isn't 20 millimoles enough for 100,000 years?
Well, that's where we get into this question of, is that concentration with that probability with other things going on for a billion years or however many years you want, is that enough? And you accumulate products. Sorry?
You only need 10 to the 20 nanoseconds.
Well, I'm not actually arguing because I don't know the answer. All that I'm saying is that if you look at the pieces that you require to have the whole thing go, you have to have a number of things happen and then come together. And it's that coming together part that I have a problem with. That's why it wasn't chaos, that's right. And if there are principles in that which make it non-chaotic, which organize themselves in a way that gives us a sort of a pathway to self-assembly, then I think we've got a grip at that point on what goes on. And I'm enthusiastic about anything that leads in that direction.
I'll give you just one possibility.
Yep.
Take nitric oxide, turn it into nitrate in the ocean. You put it to ferrous oxyhydroxide, and you can convert it 8 electrons to ammonium within 3 hours. It's almost a biological reaction. And those reactions can happen in that same place. Other reactions can interfere with that reaction, but you can certainly make amino acids out of it.
Yep.
You can start from the bottom.
So I think you can make amino acids. I think you can even make simple sugars. What I don't see is how you make a metabolic pathway. And this may be my lack of imagination or cardiovascular, oncoming cardiovascular disease, but I just don't see how you do it. It's not in principle I don't see. In principle I see it's the statistics and the probability of putting it together in one place at one time in such a fashion that it becomes self-sustaining. And so I don't disagree with the principle, I just don't see the experimental demonstration that even suggests it could happen.
Yeah, I'm not a chemist, but I really enjoyed
Who is
here.
asking the question?
Here.
Ah, oh, Oh, there. Okay.
Yes. I'm not a chemist, but I really enjoyed the introduction. It was very thought-provoking. But I wanted to return to the slides that you showed where you had some chemical reactions, and you either could have a flat line or you could have an oscillatory behavior. Right. Now, oscillatory behavior means that it's periodic and it's bound, right? But the nature of chaotic motion is that you can have oscillations, but they're non-periodic. And the other thing is that many chaotic systems are able to explore all phase space, and these bound systems do not. So the question that I have is, are you able to produce in your laboratory some reactions which you can classify as chaotic? For example, you have oscillatory behavior that doesn't have a fixed periodicity, and can it perhaps then eventually transform itself into some sort of thing where it explores all phase space that you give it?
That's a really interesting question. The answer is yes and no. And what I mean by that is that if you take the system that's cleanly oscillating and you do the laboratory experiment of pushing it to the point where it's about to undergo some bifurcation, at that point it becomes irregular. You have this phenomenon that you're probably very familiar with known as critical slowing down. And the system, other stuff starts to happen. And if you look at a typical folded bifurcation in that dotted line region, you don't have stable state solutions, which means that any perturbation from the outside flips it up or flips it down, which isn't exactly the same thing as chaotic, but it's non-periodic. I think you can make that happen, but I don't think that is truly chaos in the sense that you're talking about it. On the other hand, when you think about the motion of something in a, cycling around this pathway trajectory in a strange attractor system, the stuff that happens there is sometimes hard. It's hard to tell that you've actually got an attractor of any sort as opposed to a lot of random motion. So I think those are all the right kinds of questions, but I don't think we can answer them yet with this system, which is why I say yes and no. So I don't know. Maybe I should have just said I don't know.
Okay, there's so many really wonderful tidbits in there. But the one point that I wanted to make was when you were talking about basically having the inside of the cell stirred. You were warned by your mother, we biologists are warned that chemists only see cells as bags of molecules with a stirring bar. But if you look at, like an amoeba, you've got the gel salt state. Any cell, there's partitioning, and the more we learn about it, the more we see the partitioning. And so I'm wondering if you could buy it an awful lot by starting to look at it as eddies and currents and backwaters rather than a uniformly stirred cytoplasm.
Again, let me say two things, yes and no. The no is that you don't have eddies and backwaters in something the size of a cell because you can't get enough momentum conserved, that the momentum doesn't allow you to have an eddy before it's dissipated by friction.
Right.
But on the other hand, what you see in modern cells is you see motors everywhere. And these motors everywhere, we think of them as carrying cargo, but they probably serve other functions as well. So actually the inside of a cell may well be stirred more actively than we think.
Right, but it's more than that. I mean, I primarily worked with eukaryotes, but you would get enclosed nuclei and so on, which are, like rocks in a river, but you certainly had things, endoplasmic reticulum and so on, even in a bacterium. So you don't get a uniform stirring. So you do.
And you've got pumps, pumping stuff out. All kinds of stuff is going on. So I don't think that we should think about the inside of a cell in any sense as being a stationary drop of water.
Right, and I think that works to your advantage, not.
I think it works to our advantage rather than anything else, yes. Exactly. We have a stirring motor, but it's not a stirring motor. It's a gazillion little perturbations that cause things to flop around. Yeah, absolutely. I completely agree. Okay. Thank you all.